Imagine a world where we can unlock the secrets of the brain with unprecedented precision. This is not just a dream. It's becoming a reality, thanks to a revolutionary technique known as CRISPR screening by AAV episome-sequencing, or CrAAVe-seq. This innovative method, recently detailed in a study published in Nature Neuroscience, is transforming the landscape of functional genomics by allowing researchers to explore essential neuronal genes directly within living organisms. By utilizing adeno-associated virus, AAV, vectors combined with advanced sequencing technologies, CrAAVe-seq enables scalable and cell-type specific CRISPR screening right in the intact brain, providing a level of detail and scope that was previously unattainable. Historically, CRISPR screens have primarily been conducted in laboratory settings using cell lines, where pooled gene disruptions are analyzed for their effects on cell behavior. While these in vitro methods have proven effective, they often fall short of capturing the intricate and diverse cellular environments found in the brain. The complexity of neuronal interactions and the unique context of brain cells are crucial for understanding gene function. Yet traditional approaches have struggled to replicate these conditions. In vivo CRISPR screening has faced significant obstacles, such as inefficient delivery methods, limited scalability, and challenges in isolating results at the level of individual cells or specific cell types. However, CrAAVe-seq elegantly addresses these issues by employing engineered AAV vectors that maintain episomal DNA, allowing for the integration of CRISPR guide RNA barcodes alongside targeted gene modifications. At the core of the CrAAVe-seq platform is the clever use of AAV episomes, which are stable pieces of extrachromosomal DNA that persist within infected neurons. This stability allows researchers to sequence AAV episomes directly from isolated cell types, eliminating the need for complex single-cell RNA sequencing or sorting techniques that can dilute or obscure the signals from the screening process. By sequencing these episomes, scientists can accurately track each guide RNA barcode within specific cell populations, linking gene modifications to cellular viability and function with remarkable precision. A key component of this technology is the use of Cas9 expressing transgenic mouse models, which provide a consistent environment for genomic editing across various neuronal populations. Researchers can inject tailored AAV libraries containing pooled guide RNAs into targeted brain regions, utilizing cell-type specific promoters to ensure that only the intended neuronal subtypes are modified. This targeted approach allows for the mapping of essential genes within specific neural circuits and cell types that play critical roles in cognition and behavior. One of the most exciting discoveries made possible by CrAAVe-seq is the identification of previously overlooked genes that are vital for neuronal survival and function. The platform's ability to conduct genome-wide screens reveals novel gene networks and pathways that are unique to neural contexts, particularly those involved in synaptic transmission, neurodevelopment, and the mechanisms underlying neurodegenerative diseases. These findings challenge existing theories that have largely been based on non-neuronal models and highlight the vast, unexplored genetic landscape that governs brain physiology. In addition to identifying new targets, CrAAVe-seq offers a groundbreaking approach for drug discovery and validating therapeutic targets. By enabling gene modifications in vivo with cell-type specificity, researchers can assess pharmaceutical candidates in their natural environments, taking into account the complex cellular interactions and compensatory mechanisms that often complicate preclinical models. The platform's flexibility also allows for combinatorial CRISPR screens, which can help unravel epistatic relationships, an essential aspect of addressing multifactorial neurological disorders. The technical sophistication of the CrAAVe-seq workflow is evident in its detailed molecular and computational components. After the AAV vectors are delivered and neurons are infected, the sorted AAV episomes undergo high-throughput sequencing to identify guide RNAs and quantify their frequencies. Advanced bioinformatics pipelines then correlate this data with loss-of-function phenotypes, enabling researchers to determine gene essentiality indices across different neuronal subtypes. The modular design of the virus allows for the incorporation of multiplex reporters and conditional elements, further enhancing the richness of the data collected. Importantly, the effectiveness of CrAAVe-seq has been validated through extensive comparisons with traditional CRISPR screens and other validation methods, such as electrophysiological assays and histological analyses. These experiments confirm that the gene modifications identified through episome-sequencing correspond to observable phenotypic changes at both cellular and circuit levels, reinforcing the biological relevance and reliability of this platform. Moreover, this methodological synergy illustrates the potential to integrate genetic perturbation data with physiological outcomes, a crucial advancement for the field of systems neuroscience. Another noteworthy advantage of CrAAVe-seq is its ability to avoid the immune responses that are often triggered by viral delivery systems. By utilizing AAV serotypes that are optimized for targeting neurons and have low immunogenicity, this method ensures sustained episomal maintenance and efficient gene editing without provoking harmful inflammatory responses. This characteristic makes CrAAVe-seq particularly suitable for longitudinal studies that investigate gene function across different developmental stages and disease progressions in animal models. The scalability of the CrAAVe-seq approach opens up exciting possibilities for exploring the contributions of specific cell types to complex brain disorders such as Alzheimer's disease, Parkinson's disease, and autism spectrum disorders. By uncovering the essential genomic elements within vulnerable neuronal populations, researchers can gain insights into the molecular underpinnings of these diseases at a level of detail that was previously out of reach. This precision will ultimately inform the development of targeted gene therapies and personalized medicine strategies for neurological conditions that have long been difficult to treat. Beyond the realm of neuroscience, the conceptual framework of CrAAVe-seq has the potential to be applied to various organ systems where cellular diversity complicates genetic investigations. The adaptation of episomal AAV delivery combined with CRISPR screening could revolutionize in vivo functional genomics in tissues such as the heart, liver, and immune system, where understanding cell type-specific gene function is equally crucial. The intersection of viral vector engineering and single-cell sequencing advancements showcased here heralds a new era in biological research. As the field progresses, the integration of CrAAVe-seq with spatial transcriptomics and proteomics technologies promises to provide a truly multi-omic perspective on gene function within the architecture of native tissues. This comprehensive atlas of gene essentiality and cellular phenotype could yield insights into developmental processes and pathological conditions with an unprecedented level of detail. Furthermore, the ability to screen epigenetic modifiers and non-coding regions using this platform expands its utility beyond traditional coding gene targets. In conclusion, CrAAVe-seq signifies a transformative shift in functional genomics by facilitating scalable, cell-type-specific CRISPR screens directly in living organisms with exceptional molecular resolution. This robust platform opens new frontiers in neuroscience and beyond, providing a powerful tool to investigate the genetic foundations of health and disease within intact biological systems. The combination of viral episome sequencing with precise gene editing is poised to have a profound impact on biomedical research and the development of innovative therapies. Looking to the future, the integration of CrAAVe-seq with humanized models and clinical gene editing platforms could accelerate the translation of these findings into practical applications, bringing us closer to the realization of personalized neuromodulation and gene therapy. By shedding light on the gene networks that are crucial for neuronal health and function in living brains, this technology paves the way for a deeper understanding of the genetic complexities of the brain and its disorders at an unprecedented scale.